Welcome to this lecture, Medical Parasitology, phylum of Protozoa. We have another class, Sporozoa. Sporozoa include different important species. One of the important species, hemoparasite or Plasmodium species, cause malaria. Malaria is an infectious disease caused by a parasite. Taxonomic classification is Protozoa, unicellular, subphylum Apicomplexa, class of Sporozoa, and the genus of Plasmodium that invades red blood cells and liver cells. The parasites are transferred to humans by the bite of infected Anopheles mosquitoes. The asexual cycle takes place in red blood cells of intermediate host and the sexual cycle in female Anopheles or final host. So the life cycle is complex because need an intermediate host, human or different animals, with female Anopheles, final host or definitive host. Because the parasite causes rupture of the red blood cells, so release hemozoin product by the parasite may cause malarial paroxysm, and paroxysm is a typical symptoms of malaria. It is leading cause of mortality and morbidity common in children under 5 years, pregnant women, travelers from non-endemic area, and people with HIV and AIDS. Why malaria, or why is it important in medicine? WHO shows or estimates that there are 350 to 500 million cases of malaria in the world. 5 million of people are affected every year. At least 1.5 to 2 million deaths per year, 90% from Africa. So malaria kills more people than AIDS. Currently, over 200 Plasmodium species, there are 200 Plasmodium species cause malaria, are recognized, and new species continue to be described. From these 200 Plasmodium species, at least 4 species infect humans, and the other species infect animals, including monkeys, rodents, birds, and reptiles. Plasmodium species which infect humans, we have 4 common species cause malaria in human. Plasmodium vivax, common name, or it's called tertiary malaria or relapsing malaria. Plasmodium ovale cause oval tertiary malaria. Plasmodium malariae or quartan malaria. And the last one, which are the important than the other species, Plasmodium falciparum or malignant malaria. The most important species, Plasmodium falciparum, as it is responsible for 50% of all malaria cases in the world, and nearly all morbidity and mortality from severe malaria. So it's called severe malaria, found in the tropic and subtropic area. 90% of the cases death found in the South of America and Africa. About morphology and the life cycle of the Plasmodium species, at the first, it is multiple stages, both in human and in female Anopheles, and complex life cycle. Need 2 hosts, multiple stages. About the hosts in the life cycle, first one, intermediate host, human, different animals, vertebrate host, where the sexual cycle takes place. The parasite multiply by schizogony by multiple vision. There's 2 stages in human. First one, exo-erythrocytic schizogony and erythrocytic schizogony. That's meaning in the liver, first stage, exo, again multiply by multiple vision, it's called schizogony. And second, when the red blood cells infected with the parasite, again multiply and develop in the red blood cells. Indefinitive host or mosquitoes, female Anopheles, where the sexual cycle takes place. The male and female gametocytes, the growth and develop in the gut of female anophels, and multiply again bisexually at the first, and then asexually inside the female anophels. Life stages include two different stages: asexual reproduction, schizogony in human stages. Start with the infective stage, sporozoid, migrate to the liver, growth and multiply, develop to numerous of merozoids, thousands of the merozoids from liver, and relays found in the bloodstream, and then in the red blood cells include ring stage, develop to trophozoid, multiply by schizogony or asexually multiple vision, develop to schizont, and small number of the troph develop to male and female gametocytes. That's found in human. Second stage, sexual reproduction or sporogony, when the male and female gametocyte ingested by female anophels, multiplication sexually in the gut, develop to zygote after fertilization, and then ookinete develop to oocyst, and inside the oocyst growth and multiply by asexually, produce sporozoids, and then migrate to the salivary gland. So it is multiple stages both in human and in the female of anophels. About morphology, if you look for this image, that's red blood cells contain the parasite. So the parasite remain in the red blood cells and complete the cycle inside red blood cells. During this cycle cause malaria. If you look for this slide, we have Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and Plasmodium falciparum, 4 species in human. If we look for the stages, there are multiple stages. At the first, ring stage in red blood cells develop to trophozoid, and then multiply by schizogony or multiple vision, develop to schizont, and male with the female gametocyte. All these stages found in these species, but morphological there are differences between these stages in these species. We talk about later. About the life cycle. The life cycle, we start with the infection. The infective stage are sporozoids from with the bites from female of anophels. Sporozoid migrate to the blood, and the blood stream directly after 30 minutes migrate to the liver. So the parasite disappear in the blood after infection during 30 minutes, found in the liver. That's first, or the infective stage. We start with the phase of the in the liver, or pre-erythrocytic phase, or hepatic stage, also called the tissue or hepatic phase. In this phase, when the sporozoid migrate to the liver, in the liver cell or hepatocyte, the sporozoid mature into schizont. At the first sporozoid, only one sporozoid multiplied by asexually, growth and develop to thousands of merozoids. Cluster is called schizont, remain in the liver, and then rupture to release merozoids. The merozoid, when released from liver, start in the phase of erythrocytic schizogony or red blood cell cycle. The merozoid invade the red blood cells. They grow inside the red blood cell, feeding on the hemoglobin, and develop to trophozoid, and morphological appear in a ring form, and the troph divided, develop into schizont inside red blood cells. That's found in all these species. A small number of the troph develop to male and female gametocytes. Okay, remain in the peripheral blood during because when ingested by female of anopheles, the sexual cycle or sporogony found in female anopheles. Some of the trophozoite develop into male and female gametocytes, and the sexual cycle takes place in mosquitoes or female anopheles. Gametocyte ingested with the blood meal, and then the microgametocyte in the gut of female anopheles growth to 8 filament shape exaflagell is called 8 exaflagell, the nucleus, and divided to 8 exaflagell by exaflagellation. It is web-like active motile filaments, and the female macrogametes mature and divided after reduction division, develop to macrogametes or female gamete. After fertilization by the exaflagell penetrate the macrogamete, develop to zygote, and then the zygote develop to ookinete. Ookinete, it is elongated in shape, penetrate the gut and develop into spherical oocyst outside the gut. Development of oocyst divided to 4,000 of sporozoid. So the oocyst the gut, outside the gut, multiply by asexually, develop to 1,000 of sporozoid, and then the oocyst rupture. These sporozoids migrate to the salivary gland of the mosquitoes, female anopheles, then goes on to infect another human host. That's found in female of anopheles. If you look for this graph, it is a life cycle of Plasmodium species. We start with the first stage or infective stage. If you look for the number 1, mosquitoes takes a blood meal and inject sporozoid. That's meaning the infective stage, sporozoid with the bites cause infection. So the infection start when the sporozoid are injected with female anopheles with from the salivary gland, and with the blood migrate to the liver. So the human liver stage or exo-erythrocytic cycle start when the sporozoid with the blood migrate to the hepatic cell or hepatocytes. Each sporozoid in the liver become and grow to trophozoid at the first, and then grow and divides into thousands of merozoids. After that, it is in the merozoid cluster. It's called schizone in the liver, found in the infected liver cell. Okay, that's the human liver stage or exo-erythrocytic cycle. After that, when the merozoid release from the hepatic cell, the merozoid found in the blood stream. After that, in the blood, we have a second stage or erythrocytic cycle. That's meaning the parasite found in the bloodstream, and merozoid penetrate red blood cells. In the red blood cells, the parasite feeding on hemoglobin, and then growth to first stage is called ring stage. It is ring in shape in the red blood cells. If you look for the face, it is a fish shape. It is a ring in shape, consists of nucleus with thin cytoplasm, and then develop to trophozoid. It is irregular in shape, and then the troph divided by multiple vision or schizogony, develop to schizont, consists of numerous of merozoids. That's found in the red blood cells or erythrocytic cycle. Time of the infection in the red blood cell, it is different, depend on the species, because each species time inside the red blood cell is different from the other species. The cycle in the red blood cells return many times and cause important symptom is called paroxysm of the malaria, which consists of chills, fever, and sweating. A small number of the troph develop to gametocytes. If you look for the number 7, Small number of the trough develop two gametocytes, and then male female gametocytes found in the blood. So that in human, at the first in the liver, second in the red blood cells. In the female anopheles, female of the anopheles, when the gametocytes, male and female gametocyte, ingest by female anopheles with the blood meal, migrate to the midgut of female anopheles. So the sexual cycle found in the midgut of anopheles. At the first, the male gamete sites develop to 8 by exflagellation. That's meaning the nucleus divided to 8 elongated filament in shape is called exflagell. Macrogametocytes or female gametes again develop and by reduction division develop to macrogamete or female gamete. So there is male and female gamete. After fertilization, the male gamete or exflagell penetrate the female gamete and develop to zygote. The zygote become ookinete. It is elongated in shape. If you look for the number 10, ookinete, elongated in shape, found in the midgut. Ookinete penetrate the whole gut in female of anopheles, and out become oocyst. So ookinete found in the gut, inside the gut, when penetrated the whole gut, outside the gut become oocyst. Oocyst is spherical in shape, and then multiply by asexually. The ookinete contain thousands of sporozoid. After that, the oocyst rupture, and thousands of sporozoid migrate to the salivary gland of the female anopheles. That's complete during 20 days in female of anopheles. There is important thing during the erythrocytic cycle or hepatic liver stage in human. If you look for this image, that's a cell or hepatic cell. Hepatocytes, when infected by sporozoid inside hepatocyte, the parasite multiply by multiple division, develop 2000 of merozoids. That's first stage after the infection in liver. So 1000 of the merozoid release and found in the blood stream. So only one sporozoid developed 2000 of merozoid in the first stage or liver stage in human. Mode of transmission: first, female anopheles. Mosquitoes bite. The infective stage found in the with the proboscis or the bite in the salivary gland. The infection by the sporozoid, first stage of the infection. Blood. During blood transfusion, if contain the parasite inside the blood cells. The use of contaminated needle with the blood and with the parasite, and organ transplant. Number 3, congenital transmission. That's happened in the pregnant woman from the placenta. The parasite with the blood can infect the fetus before or during delivery. There are important terms in malaria: hypnozoid, relapse, and the important one, paroxysm. About the hypnozoid and relapse, that found in Plasmodium vivax and ovale. If you look for this slide, that's hepatocyte or the liver in the first stage, in the liver stage, when the sporozoid migrate to the liver. In Plasmodium vivax and Plasmodium ovale, some of the sporozoid do not immediately undergo a sexual replication, but enter a dormant phase known as hypnozoid. So the hypnozoid found in Plasmodium vivax and Plasmodium ovale, when the sporozoid remains silent and dormant in the liver, maybe for 1 or 2 years, and then return to complete the cycle and multiply, found in the red blood cells. So the hypnozoid are the dormant after infecting liver, can reactivate and undergo schizogony at a later time, resulting in relapse. So hypnozoid, the relapse found in Plasmodium vivax and Plasmodium ovale only. About paroxysm, paroxysm consists of 3 stages, which are the important typical clinical symptoms. Cold stage or chills for 15 minutes to 1 hour, caused due to rupture from the host red blood cells, escape into blood, present with nausea, vomiting, and headache. Second stage, hot stage or fever. Max temperature can reach to 40 to 41 Celsius. Splenomegaly easily palpable. And last, by develop to sweating stage. Lasts 8 to 12 hours. Start between midnight and midday. So fever is the most constant symptoms of malaria. It may occur in paroxysms when lysis of the red cells release merozoid, resulting in chills, fever, and sweating. If you look for this slide, that's the graph of paroxysm for Plasmodium falciparum, Plasmodium vivax and ovale, and Plasmodium malariae. If you look for this graph, the paroxysm in this species is different because time of the infection inside red blood cells it is different between these species. For example, in Plasmodium falciparum, the cycle inside red blood cells complete during 36 hours. If you look for the graph, the merozoid in the blood found in the first day, found in the bloodstream before infects red blood cells. After that, after the infection, the merozoid inside the red blood cell develop to ring stage, trophozoite stage, mature troph, and then multiply by schizogony, develop to schizone, contain a numerous of merozoids, and again the merozoid release in the blood. That's found during 36 hours. So time of the infection cause paroxysm when the merozoid and the red blood cell develop and complete the cycle, and then the merozoid release found in the bloodstream. That's found between during 36 hours. Cause paroxysm, as I mentioned, consists of chills, fever, and sweating. The paroxysm or time inside red blood cells in the Plasmodium vivax and ovale found between first and third day or 48 hours. If you look for the graph, merozoid found in the bloodstream and penetrate red blood cell, develop to ring stage, trophozoid, mature troph, schizone, again merozoid spread in the blood. So time of the infection or to complete the cycle inside red blood cells, it is longer than found in Plasmodium falciparum, during 48 hours. Okay. In the last species, Plasmodium malariae, if you look for the Plasmodium malariae, time of the infection it is longer than the other species, between first and fourth day or during 72 hours. So the parasite in Plasmodium malariae growth and complete the cycle in the red blood cells slowly, and that's the graph for the paroxysm or the cycle in red blood cells. Regarding clinical differentiation of the malaria, there are different differences between these species depending on the first one, incubation period, age of the cell-infected red blood cells, because young or mature red cells infected by the parasite, or number of the parasite per cubic millimeter, and number of the merozoid in red blood cells, and then cause, type of the malaria or initial paroxysm severity. At the first, Plasmodium falciparum, if you look for the Plasmodium falciparum, Incubation period is shorter than the other species. It's between 7 to 14 days, while the other species, for example, the incubation period in Plasmodium malaria is between 13 to 40 days. Red blood cells or age of the red blood cells infected by the parasite. For example, in Plasmodium falciparum, young with the mature red cells infected. So 10% to 40% of the red blood cells infected by the Plasmodium falciparum. In Plasmodium vivax and ovale, only the young red blood cells infected. In Plasmodium malariae, only the mature red blood cells infected. Number of the parasite per cubic millimeter, because the incubation period it is shorter, and all types of the red cells infected in Plasmodium falciparum. So number of the parasite, it's about 50,000 of the parasite found per cubic millimeter. While in the other species, like for example in Plasmodium malaria, number of the parasites about 6,000 per cubic millimeter. Number of the merozoid in infected red blood cells in Plasmodium falciparum, it is between 8 to 32 merozoid in each infected red blood cells. In Plasmodium vivax, between 12 to 24, Plasmodium ovale 6 to 14, and last one, Plasmodium malariae 6 to 12 merozoid found in the infected red blood cells. So Plasmodium falciparum, because the incubation period shorter, all types of the red cells infected, number of the parasite higher than the other species, number of the merozoid in each red blood cells, infected red blood cells again higher, so cause severe or malignant malaria. So Plasmodium falciparum, it is important species from the other Plasmodium species. About the pathogenesis and clinical disease in Plasmodium falciparum, because it is important than the other species, we start with the Plasmodium falciparum. Mature trophozoid and schizone are rarely seen in peripheral blood because they are migrate to the deep capillaries, found in the deep capillary, troph and schizone, which are the important stages in the red blood cell cycle, found in the deep capillary, and from the deep capillary may affect any organ, including the brain, kidney, heart, lungs, liver, spleen, or gastrointestinal tract. Rapidly produce daily chills, fever, and sweating, because time of the infection in the red blood cells shorter, and develop to severe nausea, vomiting, diarrhea. In cerebral malaria, because the parasite found in deep capillary, schizone, and troph, cerebral malaria is one of the common pathogenesis in the brain when developed to injury to the brain that can result in coma and death. Capillary blocking by a large number of red cells or lysis from an adhesion of infected red blood cells with each other and endothelial linings of capillaries develop to obstruction of small blood vessels, because the schizone and troph found in the deep capillary, and deep capillary is small blood vessels, and develop to anoxia of affected organ. One of the important disease is renal disease or black water fever, when the kidney infected with the parasite, common in Plasmodium falciparum, develop to acute massive lysis of red blood cells marked by high level of free hemoglobin and other products found in the blood and urine. So the product, when lysis by the parasite inside red blood cells, found in the blood and urine change to dark color with fever. Normal urine from yellow change to dark color, so it's called black water fever. Include fever, jaundice, vomiting, renal failure, the immediate cause of death. Liver and spleen, both of them develop to liver hepatomegaly and splenomegaly. So liver involvement is characterized by abdominal pain, vomiting of bile, hepatomegaly, enlargement of the liver, severe diarrhea, and rapid dehydration. And the spleen, splenomegaly syndrome, spleen normally enlarges during attack and then regresses in certain cases. Splenomegaly persists, causing discomfort and anemia. That's common or severe type of the malaria found in Plasmodium falciparum. In Plasmodium vivax, ovary, and malaria, it is different because time of the infection in these species it is shorter, so cause mild and moderate type of malaria. For example, fever, classically found every 48 hours because time of the infection inside red blood cells develop during 48 hours. Invades only young, immature red blood cells. Infected red blood cells are usually enlarged and contain granules, it's called Schöffren granules, inside the infected red blood cells. The liver and spleen gradually enlarge, hepatosplenomegaly, and anemia inside or develops slowly because time of the infection is longer. And in Plasmodium vivax with ovary relapse, it is common when the parasite remain in the liver and the hypnozoid. In Plasmodium malariae, type of the malaria called mild symptoms, mild malaria, because time of the infection is longer, every 72 hours the cycle complete and cause paroxysm, and can infect only mature red blood cells. Anemia and jaundice, both of them are common in malaria. First one, anemia due to breakdown of red blood cells, particularly occur or common in Plasmodium falciparum, and anemia can develop rapidly due to profound hemolysis because time of the infection inside or the cycle in the red blood cells it is shorter, so the anemia develop rapidly. The degree of anemia correlate with the parasitemia. That's meaning increased number of the parasite per cubic millimeter, and more serious in children and pregnant. Regarding jaundice, red blood cells destruction leads to increased bilirubin level and develop to jaundice. Malaria is the most common use for jaundice in endemic area. Most often it is caused by hemolysis and hepatic disinfection, more common in Plasmodium vivax with Plasmodium ovale. In severe malaria include severe jaundice and anemia, dark urine, severe malnutrition, fast or vomiting every feeds, unable to feed, difficult breathing and palpation, pale hands, tongue, and inner parts of the eyelid, dehydration, cerebral malaria, generalized body weakness, acute renal failure, conversion or feeds with this fever. That's found in the severe malaria. About diagnosis for malaria or Plasmodium species, the important test for diagnosis is blood examination. Why? Because the parasite found in the red blood cells. So blood examination is the most important test for the diagnosis. Both thick and thin blood film. At the first, thick blood film is concentration method that collect the parasite because in some sample contains small number. May be used to detect the presence of the parasite. There is parasite or not. Only give us to identify the parasite. There is parasite or not. The thick blood film. After that, second test, thin blood film. And this thin blood film, it is important. Why? To diagnosis and identification the species, establishing species identification. Because in the thin blood film, when we want to study the parasite, morphology of the parasite, because the red blood cells and morphology of the red cells remain in thin blood film, and we need to study the morphology of the red cells with the parasite. For example, in Plasmodium vivax, that is different stages in Plasmodium vivax during a thin blood film. infected red blood cells enlarged up to times, deformed, and contain granules as called Schiffner. So if you look for the red blood cells, infected red cells can be larger in size, 2 times than the normal red blood cells. First stage, the ring in shape. Ring consists of one ring inside red blood cells, it fine and contain don't. So the ring consists of the red in shape, is the chromatin, and the thin blue color, it is the cytoplasm. Morphologically, it is a ring in shape. And then the parasite developed to trophozoid. It is irregular in shape and amoebic in shape. After that, we have schizond, when the parasite divided by schizogony. We have schizond, it consists of numerous of merozoids. Number of the merozoid between 12 to 24 merozoid in red blood cells. And in the center of the merozoid, the pigment is dark and yellow in color. That's about ring, troph, and schizond. And during the blood examination, we can identify a macrogamete and female gamete, both of them found in the blood examination. In Plasmodium ovale, the ring, it is different. It is dense and defined mass than it found in Plasmodium vivax, and developed to compact, larger inside before, and then divided into schizond. Number of the merozoid, it is between 6 to 14 merozoid. It is larger in size, and the pigment found in the center, dark and brown in color. And the macro with microgamete, again the microgamete or male gamete, it is round and oval, contained in the nucleus in the center. Or the macrogamete or female gamete, round, compact, and the nucleus found in eccentric. In Plasmodium falciparum, the mature trophozoid stage and the schizond are rarely seen in the blood film because their forms are sequestered in the deep capillaries. Only early trophozoid and the gametocytes are ordinarily seen in the peripheral blood. But in general, in the blood, in the red blood cells, morphology for these stages, in the Plasmodium falciparum, infected red blood cells contain two fine dots. The ring stage may be several in one cell. So the ring, it is different because the dot consists of two dots. Developed to compact with the cytoplasmic vacuolation, and then after multiplication sexually by schizogony, schizond contain between 8 to 32 merozoid. It is small in size, and the pigment in the center, black, found in the center. Okay, the male gametocyte and female gametocyte, it is different in Plasmodium falciparum. And the microgamete or the male gamete, it is kidney in shape, larger, elongated in shape, larger than the red blood cells. While the female gamete, it is banana in shape, and the granules on compact mass found in the center. So the gametocyte, it is different in the Plasmodium falciparum. Last species, Plasmodium malariae, the ring stage, mass inside the ring. The chromatin or the red nucleus found in the ring, inside the ring. It is different from the other species. And the troph, it is or the chromatin, it is band or elongated in shape. Number of the merozoid between 6 to 12. Again, it is larger in size, and the pigment dark brown in the center. Again, there is macro and micro gametocytes. There are other tests for diagnosis. We have fluorescent microscopy. Fluorescent dyes detect RNA and DNA that is contained in the parasite. Detection of antimalarial antibodies. The other tests for diagnosis, PCR or polymer chain reaction, determine the species of the Plasmodium. Indirect tests, when the parasite causes leukopenia, thrombocytopenia, eosinophilia, monocytosis, and increase of ESR. These tests help us for diagnosis for Plasmodium infection. Treatment. The drugs of choice are hydrochloroquine sulfate, indicated for treatment due to Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale, and Plasmodium vivax. Primaquine to prevent relapse both in Plasmodium vivax and Plasmodium ovale. And thank you for listening.